Okay, so coming to DNA replication, we'll go a little fast since you already know about it, right?
Yeah.
What type of replication is it?
I forgot.
Semi-conservative,
Yeah.
right? What is the meaning of semi-conservative? Semi-conservative means one strand is an old strand and the other strand is a new strand. Got
Mm.
it? Okay.
Okay.
So there was a lot of confusion before, before they discovered that what exactly is the type of replication. People had a lot of issues with it, right? Some people thought, okay, it is non-conservative. Some people thought it is fully conservative, right? But then later on, it was discovered that this is semi-conservative replication. Okay. So what is replication? Basically, we are making a new strand of DNA. The red one is the new strand of DNA using the nucleotides which are present in the original DNA strand. The blue one is the original strand, right? So the blue one acts as a template strand, right? The template guides the new strand, right? So that there is complementary binding. Okay.
Okay.
So DNA replication is just DNA making a new copy of itself, right? And it conserves half of the original DNA, right?
Mm.
Now, what is the point of retaining this one strand? Is it useful?
So what's useful again? Templates?
What is the point of retaining the one strand? We are retaining one strand, aren't we? We are making a
Mm.
new one and we are retaining an old one. So why are we retaining a one?
So it can act as a template
It can act as a template. Okay, why do you want the old one to act as a template?
because well because if we have a good DNA strand that doesn't have any disadvantages DNA strands, I mean sorry, genes,
Mutations.
right, mutations, then that's a good thing. We want
See,
to keep it that way.
it ensures genetic continuity. That is a good term to use always, right?
What's
Genetic
genetic continuity?
continuity means that you have accumulated all the genes of your family, right?
Okay.
You resemble your parents, you're resembling your grandparents, right? So
Yeah.
there is a continuity going on in your lineage,
So
right?
the advantage is you can pass down advantageous genes.
Yes, not advantageous genes because it's
Okay.
not going to be selective. We just call it genetic continuity,
Yeah,
right?
okay.
So there is genetic continuity between the generation. That is point number one, right?
Mm.
Uh, what also it does is it ensures that cells, see, generally what happens, now cells in our body, they are regularly replaced, right?
Yeah.
Everybody has a fixed lifespan. Lifespan is over, it has to be replaced, right?
Mm.
So we need new cells to replace the old one, right?
Yeah.
So replication, cell division aids that process very easily, right? You're getting
Yeah.
your mother's genome, you're getting your father's genome, it aids it and you get what you want, right? So DNA
Mm.
replication occurs as a prep for mitosis, right?
Yeah.
You understand what is mitosis?
Yeah, it's a subdivision.
Cell division. How is it different from meiosis?
Mitosis, it's used for growth and repair. It's one cycle cell division.
Mm-hmm,
So the cell just divides into two identical daughter cells. The
mm-hmm.
difference between mitosis and meiosis is mitosis has identical cells which have 23 pairs of chromosomes, while in meiosis it's gametes that are produced which have 23 what is it? 23 chromosomes.
We call it diploid and haploid, right?
Yeah,
So
haploids.
mitosis is going to be diploid, right? The number of DNA in the parent molecule has to be doubled first, right? Mitosis, basically what you're saying is, I'll produce identical copy of this cell. So first you have to duplicate what is there inside this cell, right? If you want to make identical copy, you got to duplicate it,
Yeah,
right? Okay. So if this is 46, then only this will be 46, 46. So you have to double it, right?
yeah.
So that is what is DNA replication, right? It
Mm.
occurs during which phase of mitosis?
I forgot the phases.
S phase, synthesis phase, right? Cell cycle, it runs in phases. So
Mm.
DNA replication occurs during S phase. Are we clear so far?
Is it S phase?
Synthesis phase, yes.
I don't remember using that. So I think it was like we had anaphase
That
and
is stages
metaphase.
of mitosis. I'm not talking about stages of mitosis.
Oh,
Prophase, metaphase, anaphase, telophase are
yeah,
stages of mitosis, right?
yeah,
But before you enter into mitosis, you have to synthesize the DNA, right? There is G1, there is G2. G1, G2 are gap phases, and then there is S phase, where the DNA is synthesized. Then there is M phase. M phase
yeah,
is the mitosis phase. This is where cell division happens. But before cell divides, you have to make sure you have enough to give to them, right?
yeah.
It's like you've got a house right now. I'm telling you, I need two houses which are exactly the same as your house. So first you have to buy all the materials, right?
Mm,
So that is what the cell is doing. Cell is making sure it has enough so that it is able to do the division, right?
yeah.
Okay. We know the basics, right? What are the bases present in the DNA?
The bases is would be adenine, cytosine,
Hmm.
thymine, uracil.
Uracil is RNA.
Oh, sorry, in DNA, yeah. So remove that. I thought you just wanted like the bases in general
You
and
have to
go on.
remember it like this. See, I'll teach you a trick. You remember ATGC. That's the
Yeah.
way, because that's the way it's meant to be. Remember the rhyme in your head. It becomes very easy. ATGC, ATGC. Okay?
Okay.
Okay. Now T is replaced by U in the RNA, right? A and T have how many bonds? Two hydrogen bonds. And
Mm,
G and C have got how many bonds? Three hydrogen bonds, right?
yeah.
Okay. A and G are purines. Okay, type. Purine means two ringed. They have two rings, right?
Yeah.
Whereas these ones, TUC, they are pyrimidines, right? So pyrimidine means it has one ring. Yeah,
Mispyramids?
pyrimidines.
Oh, okay.
Okay, pyrimidines. Okay,
Yeah.
okay. See how to remember it. It's like an INE, right? Smaller the spelling, more the number of rings. Bigger the spelling, lesser the number of rings, right?
Mm,
Okay, so basically we are trying to make another of this DNA molecule with all these bases. The backbone is of course phosphodiester bond, right? It's a nucleotide, so it has a sugar, it has a phosphate group, and it has a base, right? That is what a nucleotide is. Sugar,
yeah.
phosphate, base. Which sugar will be here?
Oh, you ask me. Okay. It's B ribose.
Deoxyribose sugar,
Deoxy.
right?
Yeah.
Okay. Now,
Yeah.
how does it work? Let us try and understand a little bit of DNA replication, right? That is what I'm going to do. So see, if we have this DNA, we want to make copy of it, right? See, this is the phosphodiester backbone. The blues represent that. These represents the luck of the ladder. So these are all your bases, ATGC. Okay.
Yeah.
Wherever you don't understand, you have to stop me. Hmm?
Okay.
So what are we trying to do? We are trying to make another copy of it. We already discussed that it's semi-conservative. That means
Mm,
one base, one strand is not going to change, right? So the
yeah.
first step is to break these bonds, break hydrogen bonds. Hydrogen bonds are generally very weak bonds, right? They're
Yeah.
just attractive forces. So it is very easy to break them. Which enzyme comes in to break these bonds? Helicase, right? There is an enzyme called helicase which would come in to break all these bonds which are present, right?
Yes.
Okay, so the helicase is going to come in. Helicase is going to break the bonds. It is going to unwind the DNA, right?
Mm.
Between the anti-parallel nucleotides. You understand the 3 prime, 5 prime and
The what, miss?
3 prime, 5 prime and
Can you explain it? Because I think I forgot.
so it is like this. See, when we to differentiate the ends of the DNA molecule, now we write
Mm.
3 prime at one end and we'll write 5 prime at the other. So the adjacent one, you'll always write opposite. So if I've written 3 here, I'll write 5 here, 3 here. Got it?
Miss, the 3 and the 5, what do they represent? The carbon?